Okay, hello everyone. My name is Rachel Lee. I'm a rising senior at Mission San Jose High School, and I'm from the new research group. Today, I'll be presenting on the synthesis of mycophenolic acid prodrugs for carboxylesterase selective release. In this presentation, I'll introduce some of the background information on mycophenolic acid and its FDA-approved ester prodrug derivative, mycophenolate mofetil, as well as its limitations. Then I'll explain the design and synthesis of our ester analogs, our experimentals and results, and share our future plans for this project. Mycophenolic acid is a natural product derived from the fungus Penicillium stoloniferum, and it has been widely used for its immunosuppressive effects, mostly for patients recovering from organ transplants in order to prevent graft rejection. In fact, it is one of 3 immunosuppressants involved in the most common maintenance regimen for transplant patients upon discharge. As shown in the protein model, MPA in green is a non-competitive inhibitor of the enzyme IMPDH, shown in blue, which normally catalyzes the conversion of IMP, shown in orange, to XMP. And this pathway synthesizes DGTP, an important precursor to DNA replication. MPA's activity is specific for lymphocytes because not only do they depend on DNA replication for proliferation and immune response, they also depend exclusively on this pathway for DGTP synthesis. They don't have a salvage pathway unlike other types of cells. Additionally, MPA has been found to be up to 5 times more potent towards type 2 IMPDH, the type most common in lymphocytes. So overall, this makes MPA a very potent immunosuppressant. It halts the cell cycle of lymphocytes at the G1S phase checkpoint, and an unwanted immune response is prevented. Our lab has observed that esterase cleavable prodrugs like Tamiflu and benazepril can generally be used to overcome poor pharmacokinetic properties. They can be cleaved to produce the active form of the drug and can be stable in the gastrointestinal tract until bioconverted by enzymes, including esterases. This has been applied to MPA in the preparation of mycophenolate mofetil, an FDA-approved ester prodrug derivative made to enhance the bioavailability of MPA by around 90%. When consumed, the mofetil is first converted into MPA and morpholines by carboxylesterase 2 in the intestines, and any remaining mofetil escapes through the portal vein into the liver to be broken down by carboxylesterases 1 and 2. However, the varying concentrations of esterases among individuals can lead to inconsistent mofetil hydrolysis rates. To address this variability, we are synthesizing a library of 13 novel ester analogs of MPA to screen their cleavage rates under HPLC with a panel of mammalian esterases. By investigating the relationship between esterases, ester tails,
limitations of mycophenolate mofetil,
we aim to tailor our analogs towards a more diverse range while maintaining the efficacy of MPA. Under HPLC, we will use a panel of 3 carboxylesterases, human carboxylesterase 1, 2, and pig liver esterase. The 2 human carboxylesterases are found in the human liver and intestines, but reportedly exhibit selective affinities towards specific structures of esters. Generally, carboxylesterase 1 is more selective towards relatively larger acyl groups and relatively smaller ester groups. So in our case, this would be the analogs with smaller ester tails. In contrast, human carboxylesterase 2 is drawn to esters with a relatively larger ester group and relatively smaller acyl group. In our case, this would be the analogs with larger ester tails. Pig liver esterase is known to hydrolyze esters at amide bonds, and due to its optimized inflammatory response, we will also be including it in our esterase assay. As you can see, our analogs consist of a variety of structures and bulkiness levels, which reflects the varying esterase concentrations across the transplant patient population. However, our initial attempts of direct esterification from MPA were not successful. Extensive screening of conditions showed that minimal conversion was taking place, and we hypothesized that this was because the acidic phenol is a competing nucleophile in the carbodiamide-mediated acid activation. To prevent this, we followed a procedure found in literature to protect the phenol using tert-butyl dimethyl chloride. This is a two-flask process in which first both the labile phenol and the carboxylic acid are protected. And because the carboxylic acid needs to be available to attach ester tails, the reaction mixture was purified and transferred to a separate flask where the carboxylic acid only was deprotected with acetic acid. When we performed this reaction, our overall yield was 35.69%. And since this synthetic pathway requires 2 flasks and 2 extractions, it is both time-consuming and uses up materials. We also found that the multiple transfers needed increased yield variability, making it harder to determine the appropriate scale for synthesizing silated MPA for our analogs. With an optimized 1-flask silation of MPA, we first protect both the phenol and the carboxylic acid, and then quench our reaction with the weak acid formic acid in the same flask to deprotect only the carboxylic acid. This synthetic route enabled us to produce a yield of 74.15%, and by only having to perform 1 extraction, we were able to save both time and resources. We could also achieve a higher and more predictable yield due to the fewer transfers of the reaction mixture, and we could make more accurate predictions regarding the appropriate scale to synthesize our analogs on. So as you can see, we were able to reproduce this reaction on a multi-gram synthesis. We recently performed a 2-gram scale of the 1-flask silation of MPA. So we continued to synthesize TBS MPA, and we began to synthesize our first MPA ester analogs. So with this monosylated MPA in hand, we first synthesized the 2-methoxyethanol analog due to the tail's simple structure, and we hypothesized that human carboxylesterase 1 will be most selective towards this analog due to its smaller tail. So first, we performed EDC coupling in the presence of DMAP and dichloromethane, and screened the conditions in which we could deprotect the phenol. We've tried HCl, formic acid, tosic acid, and all of them decomposed except for our formic acid reaction. So after we synthesize our library of analogs, we will run our HPLC esterase assay. Currently, we're optimizing a reverse phase 18C method, which should separate esters based on polarity. And this method should be able to discern the peaks of MPA from ester prodrug derivatives. Afterwards, we'll develop a standard curve of MPA by running serial dilutions and recording the area under the curve at each concentration. When we run the Mofetil and our analogs under the esterases, we will track the peak size over time and identify how long it takes for each to fully convert back to MPA. Our lab has observed similar reactivity of molecules that also have this gamma-lactone ring. Prompted by this discovery that acidic conditions can pop open the lactone ring, this prompted our interest in preparing N-methyl lactame. We would start by preserving both silyl ethers to give this bis-siloxy mycophenolate. We would then pop open the ring with methylamine, resulting in a hydroxyamide. Our lab has utilized this protocol for the preparation of other N-methyllactam, which can subsequently be methylated to mesylate, which would turn the alcohol into a good leaving group. Subsequent treatment of this molecule with a strong base would result in a 5-exotet ring closure, followed by acidic workup to give rise to the desired lactam. For our future work, we will synthesize our remaining analogs and perform our HPLC esterase assay to hopefully procure a wide range of cleavage rates. And we'll also observe any selectivities between esterases and ester tail structures. As a future project, because MPA is associated with unpleasant side effects in the digestive, circulatory, and nervous systems, we aim to attach a thiol and a specific targeting peptide, which would allow this molecule to become more easily endocytosed by specific protein receptors. Once endocytosed, reduction of the disulfide bond would occur by reducing agents found naturally in the cell. In past literature, researchers have attached immune-related tetrapeptides like tufsin to MPA. So by attaching cell-penetrating peptides, we hope to facilitate absorption and decrease side effects. Lastly, I would like to thank my project members, my advisor, Dr. New, and my group members for all of your support and advice. And I would also like to thank ASDRP for providing us with the resources and materials to conduct our research. Thank you for listening.